At the center of the cell lies its control hub, the nucleus. It stores the cell's genetic information and directs all major activities from growth to division. Surrounded by a protective membrane, the nucleus carefully regulates what enters and exits. Inside, it holds the instructions that define the cell structure, function, and identity. The nucleus, shown here in cross-section, is the largest organelle in the cell, its central control hub. It houses the cell's DNA and directs gene expression, controlling how the cell functions. You can rotate to explore the structure and zoom in to see it in detail. The outer nuclear membrane, shown in light blue, is a lipid bilayer that faces the cytoplasm. It is continuous with the rough endoplasmic reticulum, forming a direct structural link. Ribosomes on its surface synthesize proteins that are transported into the endoplasmic reticulum lumen. The inner nuclear membrane, shown in darker blue, lines the nucleoplasm side of the nucleus. Just beneath it lies the nuclear lamina, a dense network of filament proteins. This structure maintains the nucleus shape and anchors tightly packed DNA, known as heterochromatin, to the nuclear envelope. Thousands of nuclear pore complexes perforate the double membrane, each one massive, even larger than a ribosome. Built from multiple proteins, these are not simple openings but highly selective gates. They regulate traffic in and out of the nucleus, exporting RNA and ribosomal subunits, while importing proteins like histones, transcription factors, and polymerase. DNA wraps around histone proteins to form repeating units called nucleosomes. These organize into thicker chromatin fibers within the nucleus. In regions where the chromatin is loosely packed, known as euchromatin, genes are accessible and actively transcribed by RNA polymerase. Open chromatin means active gene expression. Heterochromatins appear as dense dark regions, usually clustered near the nuclear envelope. Here, DNA is tightly packed, making it transcriptionally inactive. It is anchored to the inner membrane by nuclear lamina, and regions like centromeres and telomeres remain permanently condensed. In short, heterochromatins mean inactive DNA. The nucleolus is a dense, membrane-less region within the nucleus. Here, ribosomal DNA is transcribed to produce ribosomal RNA, the core component of ribosomes. Combined with the ribosomal proteins, it assembles pre-ribosomal subunits, which are then exported out of the nucleus. A large, prominent nucleolus signals a high rate of protein synthesis. Messenger RNA exits the nucleus through nuclear pores into the cytoplasm. This follows the central dogma. DNA is transcribed into messenger RNA in the nucleus, which then travels to ribosomes for protein synthesis. Only properly processed messenger RNA, capped, spliced, and poly A-tailed, is allowed to pass through. Around 2 meters of DNA must fit inside a nucleus just a few micrometers wide, so it undergoes extreme packaging. First, DNA wraps around histone proteins, forming nucleosomes, like thread wound on a spool. These nucleosomes connect into a bead on a string structure, which then coils further into a 30 nanometer chromatin fiber. Finally, this highly compacted chromatin fills the nucleus in its functional, organized form. Watch selective transport happening in real time. Small lines pass freely through the nuclear pores. No signals, no restriction, just passive diffusion. But larger molecules are tightly controlled. Messenger RNA carrying an export signal is recognized by exportin and allowed to pass out, while proteins like RNA polymerase with a nuclear localization signal are actively imported in. This entire system is powered by the RAN-GTP gradient, making it an energy-dependent, highly selective transport process. Watch the central dogma in action. RNA polymerase reads DNA and forms messenger RNA inside the nucleus. This is transcription. The messenger RNA is then processed and exported through nuclear pores after strict checking. Once in the cytoplasm, ribosomes bind it and translation begins. The nucleolus is not the cell's brain, it is a ribosome assembly factory. Here, ribosomal RNA is produced and combined with ribosomal proteins arriving from the cytoplasm. Inside the nucleolus, these components are assembled into two subunits, small and large, which are exported separately through the nuclear pores. They only join together in the cytoplasm during translation, forming a functional ribosome.